Well, Suresh, thank you very much for your kind comments. I think he and Lynn have to leave to go back to the site visit that's going on, and I could say many kind things about Suresh as well. He's been a real friend to Vanderbilt in many, many ways, and is also someone who is superb at mentoring young scientists and has a great interest in doing that. So, to begin my talk, TGF-beta, or transforming growth factor beta, is well known as a inhibitor of cell proliferation, and it's the pathway initiated by TGF-beta is well known to be a tumor suppressor pathway. So why is it called a transforming growth factor? So I thought I would begin by giving some ancient history. And the reason it was called transforming growth factor, there were two. One, we initially thought that it was produced mainly by transformed cells. We immediately showed that that was incorrect. But the other was that it would cause non-transformed rodent fibroblasts to appear transformed in monolayer culture in a reversible manner. And that's not unusual. But what was unusual, it would cause non-transformed cells to grow in an anchorage-independent manner in soft agar. And at that time, and still, the ability of a cell to grow in soft agar was the best in vitro correlate of tumorigenicity. So, and this was also reversible. This was first reported in 1978 by Joe DeLarco and George Todaro. And their partial purification of this activity showed that it co-eluded with epidermal growth factor competing activity. So they used the EGF receptor competing activity for further purification, eventually came up with TGF-alpha, which, as everyone knows, is an EGF-like ligand. Our laboratory reported in 1981, as did Anita Roberts and Mike Sporn's laboratory, that the EGF receptor competing activity could be separated on further purification from the soft agar stimulating activity and pursued purification using soft agar stimulation. So this shows the characters that were in the TGF-beta field prior to 1985. And the photo was taken a little over a year ago. So by 1984, Anita Roberts and Mike Sporn's laboratory and our laboratory had purified TGF-beta. We showed by competitive binding assays that there were high affinity cell surface receptors. We showed first that it was a growth inhibitor. Anita had purified enough TGF-beta to give it to Rick DeRink, who at that time was at Genentech. He's now a professor at University of California, San Francisco. So Rick obtained partial amino acid sequence and used degenerate oligos to screen a lambda GT11 library. This was before PCR, let me remind you. So 1984, he had a clone, and we knew for the first time this was a unique factor, and it was different from all the others that were there. So Joan Massaguet first showed by affinity cross-linking that there were really 3 receptors. So we knew pretty much all of that by the end of 1984, and after that it became a very popular field. Well now, I'm sorry, I forgot this. Anita and I wrote a history of the discovery of TGF-beta a couple of years ago, and the book's been slow to come out, but it'll be in a TGF-beta monograph edited by Rick DeRink and Kohei Miyazono. And at the time we were writing this, Anita had metastatic gastric adenocarcinoma. And at this meeting, which she and Joan Massaguet organized, she was not feeling well and unfortunately died 3 months afterwards at the young age of 64, and she is missed. So now, this was the original one cloned, and so it's called the TGF-beta superfamily, but it's quite large and includes the bone morphogenetic proteins, growth and differentiation factor, activin, and others. And these, of course, are well known to be critically important in embryonic development and a lot of normal processes, pathologic processes. So TGF-beta field now, there are about 38 ligands, 12 receptors that are a new class of serine threonine kinase receptors, 10 SMAD signaling molecules that are a new class of signaling molecules, 22 human disease-causing mutations in genes encoding TGF-beta superfamily ligands, receptors, and regulatory and signaling proteins, over 17 models involving genetic manipulation of these same genes for these proteins. And there are about 34,000 TGF-beta superfamily papers in the literature and over 12,000 last year. So some of the unique aspects now of TGF-beta, this is not the extended family, it's beta 1, beta 2, and beta 3. The ligands and receptors are ubiquitously expressed, and I'll talk a little bit about expression using a schematic prepared by Dan Stover, who is a third-year medical student and he did the emphasis program with our laboratory. But it's expressed in a pre-pro form. It's the carboxy terminal 112 amino acids. It's the active molecule. It's dimerized by disulfide linkage. This is the latency-associated peptides. There's proteolytic processing, but the LAP remains non-covalently bound to the active part of the molecule to give you the small latent complex, which combines with the latent TGF-beta binding protein to give the large latent complex. It has an extracellular binding domain, so when secreted, it's bound to the extracellular matrix. It's freed by proteolysis, matrix metalloproteinases, plasmin, and others to give the active molecule, and the activities initiated by or regulated by TGF-beta are many. It goes from extracellular matrix regulation to epithelial to mesenchymal transition. And our laboratory has focused for quite a few years, as Suresh mentioned, on growth inhibition and tumor suppression. So this is a schematic represents an hypothesis that Bob Coffey and I came up with 20 years ago, whereas TGF-beta is an autocrine-paracrine negative regulator balancing the positive growth factors. And so in the first part of my talk, I will focus on TGF-beta and its signaling pathway as a tumor suppressor gene using new data derived from conditional knockout of the type 2 receptor in epithelial cells. In the second part of my talk, I will be concerned with TGF-beta signaling in fibroblasts, the effects that can have on epithelial cells, again using conditional knockout of the type 2 receptor in those cells. So evidence for TGF-beta pathway being tumor suppressive. There were transgenic mouse studies, some from our lab. I won't present those. But this shows mutations, somatic mutations that have been described in human cancer. And they occur in 3 of the molecules, the type 2 receptor, the type 1 receptor, SMAD2, and SMAD4. They're relatively infrequent except in 2 circumstances. One is with the type 2 receptor. If you have microsatellite instability, it's quite numerous. Non-small cell lung cancer, glioma, gastric, and colon cancer. And the reason is there is a 10 adenine track within the coding region. So with mismatch repair difficulties, you get loss of 1, 2, 3, 4, or addition of adenines, a frame shift, and loss of expression. The other circumstance in which it's common is SMAD4 or MADH4 in the pancreas, also fairly frequent there. But there are other changes besides this. There's decreased expression of both the type 1 and the type 2 receptor through epigenetic silencing. Polymorphisms have been described that affect signaling efficiency. Increased expression of inhibitors of MAD signaling, such as EVI1, SMAD7, and STRAP. STRAP synergizes with SMAD7 to inhibit TGF-beta signaling. STRAP was cloned by Prandada, who's here, when he was affiliated with our laboratory. You can have loss of expression of facilitators of SMAD signaling, DAB2, and cytoplasmic PML, amplification of MYC, activating mutation of RAS, or inactivating mutations of downstream effectors such as the retinoblastoma gene and cyclin-dependent kinase inhibitors. So if you consider all of this, I think it's fair to conclude that the TGF-beta tumor suppressive pathway is likely impaired in a majority of common human cancers, and I think this is the same as the growth inhibitory pathway. So the mechanism of suppression, I think, is cell cycle checkpoint, and the G1S checkpoint in part. TGF-beta in some systems promotes apoptosis, and others it's a survival signaling pathway. It has been reported to stabilize the genome. But as I will tell you today, I think a major mechanism of suppression of tumor formation is regulation of growth factor, cytokine, and chemokine expression, which regulates the microenvironment. For almost as long as we've known TGF-beta was a growth inhibitor, we've known that it could enhance tumor progression. And we know that carcinoma cells frequently become refractory to the growth inhibitory effect of TGF-beta. That's associated with increased TGF-beta production in the microenvironment. High local concentrations of TGF-beta can have effects that benefit tumor growth, such as suppressing immune surveillance. They're very effective inhibitors of many immune functions. You can have increased stroma formation, and they're very good secondary angiogenic agents. More recently, we know that TGF-beta signaling in the carcinoma cell, even though they're not growth inhibited to any large extent, can still cause decreased cell-cell adhesion, increased motility, and EMT changes that would favor invasion of metastasis. Now, if these last three effects are important, then you would expect those tumors that I told you about, particularly colorectal carcinomas, that have mutations of the type 2 receptor would be less aggressive, and actually there are data to support that. This is from Stan Hamilton's group, and he was able to accumulate enough human colorectal cancers that either did not have type 2 receptor mutations and did, and look at the difference in survival. With type 1 receptor mutation, almost an 80% 5 and 10-year survival, but without that, where you can still have TGF-beta stimulation, it's down to 40%. So data such as this has prompted industry finally to become interested in TGF-beta and to develop antagonists of this pathway. So this is the oversimplified view of TGF-beta signaling in cancer. Loss of the growth inhibitory component of the pathway, perhaps due to retinoblastoma or CDK inhibitor loss or MYC amplification, et cetera, but you still have this. So they've developed pan-TGF-beta monoclonal antibodies. This soluble or the extracellular part of the type 2 receptor linked to FC. The type 3 receptor, which I won't talk about, but at least one of its functions is to bind the ligand, particularly TGF-beta 2, and present that to the type 2 receptor. And then more recently, several companies have developed small molecule inhibitors of the type 1 receptor, and two of these various compounds are now in clinical trials. I'm not sure it's this compound, but Lilly has a small molecule inhibitor in clinical trials, and Genzyme, a TGF-beta monoclonal antibody. Well, we were interested in this whole schematic, and wanted to create mouse models to do preclinical studies. And to explain the route we took, let me just briefly review TGF-beta signaling in a grossly oversimplified manner. The type 2 receptor is a constitutively active transmembrane serine threonine kinase. It binds ligand, recruits the type 1 receptor, transphosphorylates and activates that receptor, which then phosphorylates SMAD2 and SMAD3, which combines with SMAD4, enters the nucleus, regulates the transcription of many genes, including those involved in cell cycle arrest, such as decreasing MYC, increasing expression of the cyclin-dependent kinase inhibitors, which blocks the G1S to transition. SMAD7 again is a negative regulator. And then there are non-canonical signaling pathways. There are many more than this that affect the epithelial to mesenchymal transition and motility. So we really couldn't pick out one or even two genes encoding these proteins that would do what we wanted. So we finally decided to knock out the type 2 receptor. And Mark Takato's group showed several years ago, almost 10 years ago, that if you do it in embryonic stem cells, you have embryonic lethality at about day 10.5 due to lack of yolk sac blood island formation. So we chose the typical Cre-Lox approach, Anna-Kitzel in the laboratory, inserted LOX-P sites on either side of exon 2. So when that's excised by Cre recombinase, you end up with a premature transcription termination site in exon 3. It does work. These animals are now in use in at least 3 dozen laboratories around the world. We have used them to knock out the type 2 receptor in the 6 epithelial cell types shown here. And somewhat surprisingly, the phenotype was minimal. Now, this doesn't mean that endogenous TGF-beta signaling isn't growth inhibitory. I suspect that there are compensatory mechanisms. However, if we combine the knockout with oncogene activation or tumor suppressor gene attenuation, we have a marked effect suggesting that the tumor suppressive part of that is not compensated for by other signaling pathways. So what I'll do next is talk about the pancreas very briefly, and then the small intestine and colon, and spend more time on the mammary gland where we have more mechanistic data. The pancreas work was done by Hideaki Ojichi, a postdoctoral fellow in the laboratory, a gastroenterologist, and he used, in collaboration with Chris Wright's group, PTF1A Cre to knock out the type 2 receptor in all pancreatic epithelial cells, no phenotype. Next, he combined this with a knockout of an active KRAS. The KRAS, this is from Tyler Jackson, Dave Tuvinson. And so just the active KRAS at 7 weeks has pancreatic intraepithelial neoplasia. But if we had knockout of the type 2 receptor, by 7 weeks, the pancreas is full of ductal adenocarcinoma, looking very similar to what you see in humans. And it's multifocal. It's associated with a lot of desmoplasia, that is increased fibroblasts and collagen, more so for those animals that survive longer, and this shows the data. This has been, was published late last year. But this is the controls. This is with the heterozygous knockout, and there's even an effect with loss of one allele of the type 2 receptor. The work on the intestine was done by Bill Grady. I was mentor for Bill in the Vanderbilt Physician Scientist Development Program. He initiated the work here, then moved to the Fred Hutchinson Cancer Center. So the work was mainly done by graduate student Nuna Munoz in Seattle. And they used Villain-Cree to knock out the type 2 receptor in intestinal epithelial cells. Again, no discernible phenotype. So Bill decided to combine this with a weak APC mutation, the 1638N. It's weaker than the men allele. So at 9 months here, you can see that the polyps, this is the histology, this is the control, the flox alleles have no effect. But if you combine that with knockout, you get lesions that look like this. It's invasive adenocarcinoma, again with a lot of desmoplasia, and here are the data. This row is with just the APC, this one with APC plus knockout. You can see the average number of tumors per mouse is about the same. The total number, the difference is here. More adenomas with high-grade dysplasia and many more adenocarcinomas, such that the odds ratio of developing high-grade lesions for the combination APC mutation R2 knockout is 10.2 compared to just the APC mutation. The mammary gland, initiated by Anna Ketchel and then carried through by Elizabeth Forrester as a graduate student. MMTV mediated knockout of type 2 receptor in mammary epithelial cells gave a minimal phenotype, which I show here. You have on the bottom, that's the knockout lobulo-alveolar hyperplasia in virgin animals, but that goes away, indicating that TGF-beta signaling is a survival factor for epithelial cells in this context. So the next experiment was to add an oncogene, and we decided to use the MMTV polyoma middle T from Bill Mueller to get animals that have both, and this shows the percent tumor-free in a Kaplan-Meier plot with just the polyoma middle T, typical to what everyone else gets. And if we add the type 2 receptor knockout, we get tumors more quickly, indicating that TGF-beta signaling in this context is tumor suppressive. If you get rid of it, you get tumors more quickly. But we were more interested in metastases, and it actually predicted that the knockout would protect against metastases. What we got was the opposite. We had to terminate the experiment early before those with just polyoma middle T got very many metastases because these animals were very sick. That's been published. I won't present quantitative data. I'll present another model that Elizabeth worked with that has not been published, where we use the MMTV-CNU. We haven't done it with the knockout, But we have with MMTV dominant negative type 2 receptor, and Agnes Gorska in the laboratory had shown a few years back that these animals spontaneously get tumors with a median latency of over 2 years. So now if we combine the two, it takes a bit longer, indicating that TGF-beta signaling may cooperate with CNU in giving the transformation. But again, we were more interested in the metastases. So this is a typical CNU metastases. It tends to stay within blood vessels, but if you add the dominant negative, there are many more. This is quantitation. It's highly significant. So now I've shown you two models, one where we've knocked out TGF-beta signaling in the carcinoma cells and one where we've attenuated it. And in both circumstances, we get more metastases. Now, as this data were coming out, there was data coming out, particularly from Carlos Ardiega's laboratory, that if you overexpress TGF-beta, and these mutations just make it be released in an active form, it doesn't have to be activated, along with new or polyommobility, more along METs. And then Joan Massaguet's lab and then Carlos's lab again did it a different way. They used a constitutively active type 1 receptor, so they don't need to have a ligand there. Again, more metastases. That was with the CNU model. So far I've told you that in mammary carcinomas, enhanced TGF-beta signaling gives more metastases, and attenuation or loss gives more metastases. And we're not the only one that's observed that. Xiaojing Wang at Oregon Health Sciences University has very similar findings in mouse skin carcinogenesis. So how do you explain this? Well, a typical explanation with enhanced TGF-beta signaling is you have more TGF-beta induction of EMT, which allows invasion and metastasis. And then, of course, the TGF-beta effects on host cells that I've talked about. And you could have the same thing with the dominant negative because it doesn't completely block TGF-beta signaling. But when you get to the knockout, you can't explain it, or I can't. If anyone can, I would love to hear what it is, unless you bring in other cell types. And so attenuation of loss of TGF-beta signaling can have alterations in the microenvironment, and I'll talk about that. Our thinking on this was influenced by some DNA microarray studies. This is just two separate ones, and this shows fold increase in the knockout compared to the non-knockout carcinoma cells. You have a lot of chemokines that are increased in expression, some are decreased, and an orphan receptor. But remember CXCL5 here, over 50-fold, and Brian Beery did a lot of this in the data analysis. Brian's a graduate student. So Li Yang, who was at that time in David Carbone's laboratory, had been working with myeloid immunosuppressor cells, and she had a very nice paper in Cancer Cell 3 years ago, and I'll briefly review what she had shown in this paper. But these myeloid immunosuppressor cells, MISCs, also known as MDSCs, have markers of both granulocytes and macrophages. They're immature. They can make up 5% or more of total cells in the tumor, can promote tumor growth. When co-injected with tumor cells, produce tumors with increased vascularity and decreased necrosis. They produce MMP9. Deletion of MMP9 in the MISCs abolishes their tumor-promoting activity, and they can be incorporated into the tumor endothelium. So Lee's hypothesis was that our knockout cells were drawing in more MISCs that would then promote the metastasis, the increased metastasis. And so she did the first experiment. This is a flow cytometry using both GR and CD11B as markers. And you can see with the knockout there are more than in the control, and this provides quantitation, quite a big increase. So in the knockout, the MISCs can be more than 5% of the cells present. Their localization is quite interesting using GR1. Immunohistochemistry. They're not throughout the tumor, but they tend to be at the edge, and this is shown even better in another model that Lee has used. We wanted to use a model that did not have an introduced viral oncogene. So she chose the 4T1 cells that were carcinoma line that was derived from a spontaneous tumor arising in a biopsy mouse. So in those, about 7% of the cells are MISCs, and looking at the localization there, not many in the adjacent mammary gland, a few in the tumor, mainly at the invading edge or the periphery. So they not only accumulate in the tumor, but also in other parts of the body. For example, the spleen 28 days after orthotopically implanting 4T1 cells, the spleen, over 50% of the cells have these two markers. And this shows the kinetics. In the bone marrow, they're normally 20 to 30%, which you would expect for immature cells. But as you go longer with the tumor, it goes up to 80%. So Lee was able to purify to a reasonably high degree of purity, 95 to 98%, the MISCs from tumor tissue and spleen. And she then used those with the 4T1 cells. She used a ratio of 1 MISC to 10 carcinoma cells, and then orthotopically implant that in the mammary gland, and then counted metastases. And you can see that MISCs from non-tumor bearing animals have very little effect. From the tumor spleen have some effect, but tumor infiltrating MISCs have a very significant effect in promoting lung metastases. So how do they do this? Well, one way to look at it is to look at in vitro invasion assays through a matrigel coated transwell. The MISCs are, she has a red tracking dye. The carcinoma cells a green tracking dye. Here's the data. The non-tumor MISCs have very little effect. The tumor MISCs have cause a marked increase in invasion through the matrigel. And based on previous work, she suspected involvement of matrix metalloproteinases, and showed that a broad-spectrum MMP inhibitor blocked this invasion. So which MMPs? Well, it turns out that in the tumor infiltrating, the tumor educates these cells when they're there to produce hundreds of fold more MMP14 and MMP2, and 14,000-fold increase in MMP13. So we have a lot of MMPs being produced in the tumor by these MISCs, and we know from the work of Len Matrisian and many others, these can influence invasion and metastases. The MISCs also express a lot of TGF-beta, and this TGF-beta 1 immunohistochemistry indicates that actually most of the TGF-beta is being produced by these myeloid cells in the stroma, much more so than the carcinoma as shown here. So how do they get there? Well, remember I stressed the increase in the microarray of CXCL5 at the RNA level. This shows protein level by ELISA, looking at the floxed carcinoma line compared to the knockout, a marked increase. That's also true in the CNU versus the CNU plus the dominant negative type 2 receptor. So here is data showing that CXCL5 is a chemoattractant for the MISC cells, significant. The conditional medium is a good chemoattractant, and a neutralizing antibody to CXCL5 inhibits that significantly, and this is just the control here. We also developed evidence for another chemokine receptor involvement, and that is SDF1 or CXCL12 and the receptor CXCR4. The expression of SDF1 doesn't really change with the knockout as compared to the control. However, as shown here, the tumor infiltrating MISC cells acquire expression of CXCR4 both in the polyoma middle T model and in the 4T1 model. Again, functional data showing that SDF is a chemoattractant for the MISCs. It's blocked by neutralizing antibody to CXCR4, the receptor. And of course, there's no effect with the control ones because they don't have the receptor. So to summarize what I've told you in this segment, loss or attenuation of TGF-beta signaling in carcinoma cells enhances metastasis. It's associated with recruitment of MISCs into the tumors at the edge or the invading edge. These cells promote carcinoma cell invasion and metastasis. MMPs and TGF-beta secreted by MISCs appear to be important in promoting the invasion and metastasis, and the mechanism of recruitment involves two chemokine receptor axes. So to now sort of wrap up our working hypothesis now using the schematic again with knockout of the type 2 receptor and oncogene activation or tumor suppressor gene attenuation, increased expression of chemokines, growth factors, cytokines associated with a desmoplasia, likely due to this influx of bone marrow-derived cells, and we think the influence of that on the epithelial cells accounts for the increased growth, invasion, and metastasis. Now, I want to stress that these immature bone marrow cells are not just in mice, and I'll present this slide from a paper published by David Carbone's laboratory 7 years ago. And let's just, you have to use different markers for the immature bone marrow cells or the MISCs in humans, and this is in peripheral blood, and this is a control. This is a stage 1 to 2 carcinoma patient, and this is a stage 3 to 4. And you can see that the number of these circulating in the peripheral blood goes up, and that holds for other markers. It also holds for lung cancer and head and neck cancer. And one other thing on humans, work that Lee did, if we looked at 8 human adenocarcinoma of the breast at bone marrow-derived cells, and we found them in all of these at the invading edge. So now let me turn to the effect of knockout of the type 2 receptor in tissue fibroblasts. This was done by Neil Bomick when he was a graduate student in the lab. Neil now has his own independent lab here at Vanderbilt. It was done in collaboration with Eric Nielsen, who had developed the FSP1 Cree mice. And before showing you the striking epithelial phenotype we got in these, let me show you where they're expressed. And this is, of course, crossing the FSP1 Cree with Rosa 26 reporter mice. 2 points. The Cree is expressed in the stroma, not in the epithelium, and the 3 organs I'll talk about. And it's not expressed in all of the fibroblasts. You see a number of the blue ones here that have expressed it, a number of fibroblast-like cells that haven't. You see that over here as well. So first I'll show you the prostate phenotype. This is the dorsolateral prostate at 6 weeks of age. And the knockout cells, by 6 weeks, 100% of the animals had epithelial hyperplasia with the tibia, in other words, pancreatic intraepithelial neoplasia, and an expansion of the stroma. If we use phospho-SMAD2 as an index of endogenous TGF-beta signaling, you can see that in the control, you have, in the brown, evidence of TGF-beta activity in both the epithelial and the stromal cells. Here we retain TGF-beta signaling in the epithelial cells, but it's lost in a majority of the stromal cells. Ki67 staining, we have increased proliferation in both the epithelial and stromal component. Next is the forestomach. This is the esophagus here, the forestomach, the glandular stomach. This is an H&E section from that junction showing the stratified squamous epithelium lining of the forestomach, the glandular stomach. Again, 100% of the animals by 6 weeks of age had tumors in the forestomach, which were invasive squamous cell carcinomas. And again, using phospho-SMAD2, you can see staining in the control here in both epithelium and stroma. And here you have staining evidence of TGF-beta signaling in the carcinoma cells with loss in many of the stromal cells. Now, the mechanism that we suspected from earlier studies in our laboratory was increased hepatocyte growth factor expression by the knockout cells. And Neil showed that. This was published 3 years ago. I won't stress this, but both in the forestomach and the prostate. And there was also increased activating phosphorylation of the cognate receptor, c-Met, and the knockout here, there, and there, compared to the 3 controls. I'll now show you some unpublished data done in collaboration with Felix Chu and the immunohistochemistry team at Exelixis, and they had developed a very good phospho-Met immunofluorescence assay. And you can see here with the knockout, there's in the red a marked increase in phospho-MET. It's in the epithelium as expected. Now, the reason we were collaborating with Exelixis is that they had a small molecule inhibitor of c-MET that they were willing to share with us, XL7592. And this part of this work was done by Nikki Ching. I'll talk about her later, and by the group at Exelixis. But you can see that a 13-day treatment with XL7592 results in almost complete inhibition of phosphorylation of c-MET, doesn't bother total MET. And the same thing was true for RON. RON is a c-MET-like receptor, heterodimerizes with it, and you can see inhibition of phospho-RON. So we've inhibited the target. Did it have an effect? And the answer is yes, sort of. So this is a Ki-67 staining. You see a decrease as we go over this 2-week period. Tunnel staining for apoptosis, an increase. Put the two together, it becomes a little more striking. But if you look at the histology on the bottom, these carcinomas are progressing. So other things are involved. If you look at the very earliest lesions here, before there's much change in the epithelium, you have an influx of these mononuclear cells using the panleukocyte marker CD45. They're positive, so we have an influx of the bone marrow-derived cells. If we look a week later, where you have quite a lot of epithelial atypia now, you can see that they remain intimately associated with that. So based on Lee's work from many other laboratories, we suspect that this influx of bone marrow-derived cells is helping drive this carcinogenic process. Mammary gland tumor studies. So how do you study mammary gland tumors in mice that die by 8 weeks of age before the mammary gland really develops very far? And the answer is you check with Simon Hayward, who can teach you how to do tissue recombinations, which we did using polyoma middle T or 4T1 cells, combining them with either wild type or knockout fibroblasts, and collagen, put them under the renal capsule, and then harvest at whatever time. And this is work done by Nikki Ching. One paper's published, another's in press. And in the interest of time, I'll just summarize this work. With the knockout fibroblasts, they had larger primary tumors with increased invasion, increased metastasis to the lung, liver, and spleen, increased expression of hepatocyte growth factor, macrophage stimulatory protein, which is the ligand for C-RON, and TGF-alpha. We have increased phosphorylation of the cognate receptors on the tumor cells. And Nikki showed that treatment with the small molecule c-MET inhibitor decreased tumor size and the number of metastases. And siRNA knockdown of c-MET decreases metastases. So that's, I think, compelling evidence for the HGF-c-MET axis and the pathogenesis of, the lesions that are enabled by the knockout fibroblast. But more is going on. Nikki has now, of course, shown increased chemokine expression in the knockout fibroblasts, increased macrophage infiltrates in these tumors, and is pursuing the involvement of chemokines and bone marrow-derived cells in the processes that we've been talking about. So now to give sort of a summary and working hypothesis for the knockout in the type 2 receptor in fibroblasts. You have increased expression of HGF, MSP, TGF-alpha. This could have an effect on these cells, although loss of TGF-beta growth inhibition could help explain the increased desmoplasia that we see wherever there's an epithelial lesion. These have increased expression of secretion of chemokines. We have more bone marrow-derived cells, and I think the combined influence on the epithelial cells can account for the carcinoma in situ and invasive carcinoma that we saw in the animals, or the enhanced carcinoma growth, invasion of metastases in the tissue recombination experiments. So in conclusion, TGF-beta signaling in epithelial cells is a tumor suppressor pathway both early and late in carcinoma development. TGF-beta signaling in tissue fibroblasts is tumor suppressive for adjacent epithelia. Mechanisms include both cell cycle regulation and regulation of expression of growth factors and chemokines, and the chemokine recruitment of immature bone marrow-derived cells can play a major role in carcinoma progression, and very importantly, present new targets for therapy. So let's come back now to the question of whether clinical trials in cancer patients using inhibitors of TGF-beta signaling is likely to be worthwhile. Well, in our model, probably not, but I'm sure there may be some circumstances where this sort of schematic works. But I think more importantly, we should use these to block TGF-beta suppression of immune surveillance. It's been my sort of maybe biased opinion for over a decade that the reason for failure for virtually every immunotherapy or vaccine trial is the failure to take into account what the tumor is doing to block that effect. So now with these, there might be a way to do that, and TGF-beta certainly is a major one. VEGF is probably another, but there's an approved drug for that on the market now. And another circumstance is this vicious cycle in bone metastases, as demonstrated by Greg Mundy, who's a new faculty member here, heads the Bone Center. He's a part of the Tumor Microenvironment Grant with me and people in my laboratory and Simon Hayward. So we're beginning to talk about this, and I think that may be another circumstance. So I've given credit to the people in the lab who've done the work as I've gone through. I did want to mention that Mary Ockery, Anna Kitchell, and Agnieszka Gorska contribute to all the studies. They are the permanent members of the laboratory. Our collaborators, I mentioned Neil Bomick, David Carbone, Charles Lynn, and Lynn Matresian collaborated with Lee on the bone marrow-derived cell things. I mentioned Bill Grady, Simon Hayward, who helped with the tissue recombination, Eric Nielsen with the FSP Cree mice, and the collaboration in the knockout. You sure did all of our biostatistical analysis, and Allison Jolie at Felix Chu at Exelixis. Allison provided the small molecule inhibitor, CMAP, and did the pharmacokinetic studies, and Felix, the immunohistochemistry that I showed. So here's the crew in the laboratory. Since I have a minute or so left, let me mention that the lab also includes Ty Abel, an assistant professor of pathology, a neuropathologist, who's a member of the Vanderbilt Physician Scientist Development Program. Kara Clark works with him. We have Pete Clark, an assistant professor of urologic surgery, and Dina, who works with him. Jonathan Shu, research assistant professor of cancer biology, and Boyana, who works with him. And this is Li Yang, Nikki Chang, and then Brian Beery, Hideaki Ajichi, who will be returning, much to my sorrow, to Japan next month, and Brian Beery and Meta Erdogan, who are graduate students. Thank you for your attention. I did leave time for questions if there are any. Okay. I was concerned there for a moment. Yes, Dan. That's what we would really like to know, but no one's looked at it. We have been using immunohistochemistry to look at it, and actually if you use the data set, or the set of slides that Helene Scobie and David Page and Bill DuPont used for a paper published a couple of years ago, I've reviewed those, and there does appear to be, in most of them, this is type 2 receptor immunohistochemistry, decreased staining in the fibroblasts relative to the control. We're now culturing human fibroblasts from human breast cancer and looking at TGF-beta signaling there. I think we also need to look at chemokine expression by those cells as well to see whether this may play a role in human cancer. And I think it's an important question to answer. But no one else has studied it, to my knowledge, except by immunohistochemistry. Yes, yeah, Jim.
I'm interested, you mentioned the bone marrow-derived cells as a new target. Do you think that, in fact, that conventional chemotherapy is affecting anything not otherwise?
Oh, where is Lee? Oh, there she is. Can I mention gemcitabine? Or? So, Lee's done one, experiment or set of experiments with gemcitabine, and actually these do decrease. Gemcitabine does decrease the number of these cells in her model or our model. And as far as any other chemotherapeutic agent, we don't know. Has anyone looked at that, Lee, that you're aware of? No. I might comment, Jim, that I've tried to avoid immunology all of my career. And I just found that I couldn't do it anymore. Was there another question over there? Okay, well, thank you very much.